the beginning of the report, it talks about how we were bringing people together to talk about bees through the Northeast, looking at, we had a pollen processing and ID class that we did. We said, oh, it'll be really easy. This is really a tough thing to do. It takes hours and hours to process the pollen with all sorts of nasty acids, and then it's really hard to identify them. So we spent 4 days trying to learn that. We have a big grant.
Can you explain a little bit about that for us? When we process the pollen?
Okay, so pollen is produced by the male part of the flower, and it's in order to withstand conditions in the environment, it's covered, it's a very tough, protected unit. And so in order to see the sculpturing and to be able to actually look at it under the microscope and tell it from different kinds of pollen, you process it to remove all of the outside material so that you can see the structure of these cells. And so that's what's going on. And so you use one acid after another in order to process them. So in order to do that. And then all the pollens in a family of plants look the same. So all of the asters look the same, all of these swamp plants look the same. Cranberry looks like blueberry, looks like rhododendron. So you have to be really expert doing that. So it's not as easy as we thought. Not anywhere as easy as we thought. And so then we brought our research group together we have researchers in the New England states and New York, where we're asking questions about bee health and what bees are in the different crops in New England. And then we also we entertained the habitat, the bee habitat working group, which is a northern New England group, so we had that. The other thing is Andrea Kudo in my lab put out this lovely product that you may or may have not seen. That it's hard copy, but it'll go up on the web also. So this is what we find is that people come up to us and they say, we want to know everything about bees, and it takes a long time to discuss it. So what Andrea did is everything you would want to know about bees. So you can just hand this to someone, which is very efficient. And so this is the one that you have is the Northeast Crops Edition, which we did for our because we have cooperators in pumpkin apple and lowbush blueberry. So that's where you see that. And then we're hoping to do one on in more urban settings, urban landscape settings. So we did that. Then we also have a pollinator garden that Marty Sylvia in my lab has been working on, so that you can come and see what plants grow here in southeastern Massachusetts and that are visited by bees in case you wanted to improve habitat. In research, there were, under the pollinators umbrella, we had 6 major prongs going. 4 of them had to do with pollinator health. And you'll see that there's a theme of bumblebees everywhere. So if you think of the glaciers receding and all of these bogs being here that have these particular plants, cranberry and relatives, there are these difficult flowers and they're partnered with bumblebees. Bumblebees are the bog, pretty much the bog pollinator. They're really common. So that when agriculture moved in and put cultivated plants in there, bumblebees are still the #1 pollinator that we see in cranberry. It's a difficult flower that needs the particular behaviors and size of the bumblebee in order to access the pollen. So you'll see that what we did, we had a project looking at what happens if bumblebee colonies, which you can get in the mail. You can just say, I want some bumblebees, and you say, send them to me, and these bumblebee colonies arrive from Michigan. We took those bumblebee colonies and put them in Noel Hahn in our lab was the one who did this. It was very hard work. Put these bumblebee colonies on grower sites and on trustees or reservation wild sites that were way away from cranberry. And we found that most of them did pretty badly. So
either one.
Pardon?
Both.
On both.
Yeah.
We had 3 out of 5 sites where they were at cultivated cranberry that died immediately, but then 2 did well, so that was awful. And then at our trustees of reservation sites, they were eaten by predators. They were just taken out. Who do you think our predators were? Was it bears, skunks,
Waxworms, cockroaches,
yeah,
ants.
just everything. So that didn't work out too well. But the previous year, you should never do an experiment, but the previous year they did very, very well on the commercial bogs in comparison to non-commercial bogs. And nobody believes this in the lab, but I think that we need to think about our use of chlorothalonil. I think that that's what the issue is on cultivated bogs. Andrea Kudo in the lab wants to know when bumblebees start the year as a queen bee, the queen bee establishes the colony. And so her question is, what pollens are these queen bees using in May? They come out, it's like the spring is just broken, there's almost nothing out there, but there are those queen bumblebees out looking around for resources. And so her question is, when do these queen bees come out and what are they using for resources? And this could inform, if we wanted to do conservation, could inform that. Another health issue that we had that we were addressing was, I just started this up with a cooperator, and I'm sure you've been hearing about this, is if you think of your gut microbiota, think of your gut, it's full of all of these microorganisms. And it's a mix of different microorganisms. And research in humans is showing that these microorganisms have critical functions in digestion, in resistance to pathogens, just in everything that you can think of. It's turning out that you could imagine. It's important. Well, guess what? It's true with bees too. So this is a new area of research. And so our question was to take bumblebee colonies and see what they're got using molecular techniques, see what their composition and frequency is of the different microorganisms in the gut, and compare that to bees that have been exposed to the #1 awful compound that's getting such bad press these days, a neonicotinoid. So neonicotinoid is a nerve poison that's utilized on millions of acres all over the world, and the problem is that it moves into the pollen and nectar. And so we wanted to know what happens to the microbiota of our bumblebees when they're exposed to vanishingly small levels of this compound. So this doesn't have anything to do with cranberry growers because they're not using neonicotinoids, but it is a great question as a starting point for other points, which what I'm most interested in is how do all of the phytochemicals in cranberry pollen influence the microbiota in a bumblebee? Because I think we have preliminary evidence showing that when you're on a cranberry diet and you're a bee, I think that there are some pathogens that are reduced. So there's going to perhaps, yeah, isn't that way cool? Wouldn't that be neat? So after that bladder, the bad press. So that we would need to know, we need this microbiota baseline, and then we can have the pollen information, and then we can see how the bees are doing. So it's health, so I got to speed up, okay. And then we were looking at, we did a preliminary test to see if you have a bunch of cranberries and you throw a tent over it and add a whole bunch of bees, do you get a higher yield? This is the Linda Renta question. And and sometimes we did and sometimes we didn't. So we either need to expand that or just more bees, put it to rest, you know, and what are we going to do? Bring in more honeybees. And so we had a couple interns also working on honeybee health, doing a survey around the state to see what pathogens were most prevalent. We had hoped to compare backyard bees with the migratory bees, but we didn't get enough samples from migratory bees to say much about that. But what we did find is that the backyard beekeepers that are having a lot of trouble keeping bees alive,
We're hearing it.
Pardon?
We're hearing it.
Yeah, they were full of varroa mite, and not the microsporidium, but varroa mite was very high. So for most of them, so there's issues in beekeeping. They want to blame neonicotinoids, they want to blame migratories, but in the end, I think that we just need better beekeeping, but don't tell them I said that. Okay. Then there's the other arm of our program is on management, and we were interested in a question of, as we move in new cultivars that develop very early, how does that influence our worst pest, cranberry fruitworm? Cranberry fruitworm is this squirrely moth that gets out there just as the bogs are going into bloom, and they're waiting for fruit to appear to lay their eggs. They're going to lay the eggs on fruit. They wait until those fruits have started to size up. So here we are changing all of our bogs to these large fruiting early cultivars. What's going to happen with cranberry fruitworm? And the bottom line that Marty Sylvia wrote up for us was that of the 112 eggs that we found and the huge number of samples that we did, most of the eggs were on these large fruiting hybrids. So
If you build it, they will come.
yeah, that's right. So, I love that movie. Okay, so the other thing we've been working on is cranberry scale. This is an outbreak of a sucking insect with a hard shell over the top that's just been sweeping through the industry. I keep saying it's going to disappear and that the natural enemies are going to take it out, but not yet. So we've been working out. The other thing we have that we're very proud of is we put out, this is a Frank Caruso product. It's the compendium of blueberry, cranberry, and lingonberry diseases and pests. And so I did the insect section, took many, many, many hours, and coordinating about 40 different authors and sections. And so that was a big output. Marty, we just did our IR4, which is a very important arm, but I guess we were working on a pathway.
We were working on your favorite chlorothalonil. We were trying to get Bravo residue levels through for European export fruit, and the EU didn't like the way it was sampled 2 years ago because there was too much time between the time it was harvested and the time that the lab analyzed it. So this year we had to fast track it within 30 days of picking the analysis, and California had to determine what the residue level was. So we continue to defend Bravo, which is our best fungicide. And next year we're going to be doing a Chateau, a lovely herbicide.
So for those who aren't